Assalamu alaikum. I am Dr. Hasiba Mahmuda, currently working as an assistant professor current charge in the Department of Microbiology, Bashundhara Ad-din Medical College. Today I am going to do a presentation on the microbiology, topics of microbiology. Today my topic is the morphological structure of bacteria. Let's get started. First, we will learn bacterial structures. This is the typical bacterial cell structure. The structure showed in the middle. We can see the DNA, bacterial DNA, which is called the nucleoid. Around the nucleus, there is a cytoplasm of bacteria, which contains the essential structure called the ribosome. Besides this, there is a plasmid. Around the cytoplasm, there is a plasma membrane, and outside the plasma membrane, there is a bacterial cell wall. It is different with eukaryotic cell. Our eukaryotic cell doesn't have any cell wall. And outside the cell wall, here there is a capsule. So it's a capsulated bacteria. There is a small hair-like structures are there, which are called pilus. And also there is a whip-like structure called the flagellum. These are the basic structures of bacterial cell. Now, the bacterial structures are classified into two categories: essential structures and non-essential structures. Essential structures include cell wall, cell membrane or plasma membrane, nucleoid, ribosome, mesosome, and periplasm. And the non-essential structures include flagella, fimbria or pili, capsule, spores, plasmid, granules, glycocalyx, and slime layer, etc. This is the chart of your test book, which is the Lange review book, and you have to learn for your exam. You have to go through this chart for your examination, both for your SAQ, short answer question, and also for your oral examination. In the next slide, I will describe these structures. And first, there is a plasma membrane. This is the content. This is the phospholipid bilayer. There is no sterol. That is, exception is mycoplasma. What the function of plasma membrane? Active transport of molecules, energy generation by oxidative phosphorylation, synthesis of precursor of cell wall, and the secretion of enzymes and toxins. Then the structure is cell wall. It is the outermost component of bacteria. But there is an exception, mycoplasma, which is the only bacteria that has no cell wall. It is only bounded by cell membrane. Then functions. The functions of cell wall include: provide structural support, maintains the characteristic shape of cell. Here we can see the gram staining finding of gram-positive bacteria and gram-negative bacteria. The color of gram-positive bacteria and gram stain is purple color, and gram-negative bacteria is pink color. The color variation is due to their cell wall property. The cell wall of gram-positive and gram-negative bacteria differs from each other. That's why in gram staining finding, the finding is different from each other. Here we can see the cell wall of gram-positive and gram-negative bacteria. Both have cell membrane. This is the blue portion is the cell membrane. This is the gram-positive bacteria, and this is gram-negative bacteria. And outside the cell membrane in gram-positive bacteria, there is only peptidoglycan layer, which is very thick. In case of gram-negative bacteria, outside the cell membrane, there is another membrane called the outer membrane. And in between the cell membrane and outer membrane, there is a space which is called the periplasmic space, which contains a very thin layer of peptidoglycan. This structural dissimilarity is of gram-positive and gram-negative bacteria. Then bacterial cell wall. We have already told about the peptidoglycan layer. Peptidoglycan composed of alternate strands of two major subunits, N-acetylglucosamine and N-acetylmuramic acid. N-acetylglucosamine and N-acetylmuramic acid is linked by beta-1,4 linkage bond. The cross-linked by tetrapeptide chain attached to the N-acetylmuramic acid portion. Pentaglycine bridge connecting to tetrapeptide chain. In gram staining of bacteria, outer membrane consists of lipoprotein layer, outer membrane, and lipopolysaccharides. This is the schematic picture of the peptidoglycan structure, as I have already mentioned. In gram-positive bacteria, there is a thick layer of peptidoglycan and there is negatively charged teichoic acid on the surface. This is the picture of your textbook, which is in the Lange review book. In the left hand side, the picture is showing the cell wall of the gram-positive bacteria, and in the right side, the figure shows the cell wall of the gram-negative bacteria. I have already described that the cell membrane is common in both of them, and the peptidoglycan layer is very thick in case of gram-positive and very thin in case of gram-negative. And gram-negative bacteria has also periplasmic space and outer membrane layer. Both gram-positive and gram-negative bacteria have the flagellum and pilus, it's common. But teichoic acid is an additional part of the gram-positive bacteria, which is negatively charged. Then bacterial cell wall. Why gram-negative bacterial cell wall differs from that of gram-positive bacteria? The cell wall is much more complex in gram-negative bacteria. There is a thin peptidoglycan layer and filled and surrounded with periplasmic space. So the outer membrane is unique. And the outer membrane, there is a presence of lipopolysaccharide, which is composed of a lipid A portion and O specific polysaccharide portion. And lipopolysaccharides acts as an endotoxin. Then endotoxin, this is a picture of lipopolysaccharide. The lower portion composed of the lipid A and polysaccharide portion. The lipid A portion, which causes the toxic effect in cells, it causes the endotoxic shock, and there is polysaccharide portion, and O antigen is the outer portion. O antigen is the somatic portion, it is antigenic. Antibody is formed against this antigen in our body. Then comes to teichoic acid. It is located in the outer layer of gram-positive bacteria. It is composed of glycerol phosphate or ribitol phosphate. Importance: in gram-positive bacteria, it induces inflammation and causes septic shock. Differences between gram-positive and gram-negative bacteria. This chart is included in your book, and you have to go through this, and I have already mentioned in my previous slides. Then another essential structure is ribosome. It's very important for your exam. Function is a site of protein synthesis. Size is 70 A's. It's composed of 50 A's and 30 A's ribosomal subunit. And here the S, the letter S refers to Fedberg units. It means is the relative rate of sedimentation. The more the subunit value of S, the more will be the rate of sedimentation. And it will be more heavier. Then in eukaryotic cell, there is a difference in ribosomic subunit in eukaryotic cell and prokaryotic cell. In eukaryotic cell, it is 80S, it is composed of 60S and 40S ribosomal subunit. Prokaryotic ribosome composed of 50S and 30S, total 70S, and eukaryotic ribosome contains 80S, which is composed of 60S subunit and 40S subunit. And you have to know it for your exam, it will be asked in your exam. And here there is another point, that is selective toxicity. What is selective toxicity? Action of antibiotics on protein synthesis of bacteria only, not on human. This variation is ribosomal subunit is responsible for selective toxicity of antibiotics on bacteria only. The antibiotic protein synthesis inhibitor antibiotics does not act over our eukaryotic cell ribosome, as the subunits are different between prokaryotic and eukaryotic ribosome. Then granules. It is a storage area for nutrients stained characteristically with certain dye. For example, you have to know for your exam the metachromatic granules of Corynebacterium diphtheri. Then nucleoid. Nucleoid is the irregular area of cytoplasm where DNA or most genetic materials located. Here the DNA is single circular in prokaryotic cell. There is no nuclear membrane, no nucleolus, no mitotic spindle, no histone protein like eukaryotic cell. Then plasmids. Plasmids are extrachromosomal, circular, double-stranded DNA molecule. The plasmids can replicate independently without the help of chromosome. The plasmid can integrate into bacterial chromosome also. It is present in both gram-positive and gram-negative bacteria. Here both transmissible and non-transmissible plasmids exist. Plasmid carry gene for different functions and structures. For example, it carry gene for antibiotic resistance, such as beta-lactamase by Staph aureus, exotoxin, for example, enterotoxin of E. coli bacteria, pili, and gene carrying for resistance to heavy metals such as mercury, ultraviolet light, etc. Then transposon, it's called the jumping gene. The pieces of DNA, it's a move readily from one side to another within or between DNA of bacteria plasmids and bacteriophages. That's all for today. Inshallah, we will continue the other portion of my presentation in the next day. Thank you.